Welcome back to my YouTube channel, Life Sciences by Tumi Selane, where we share life sciences content and skills in order for you to ace your final examinations. If you like the content in my videos, please do hit me up with a like, contribute to my videos by commenting, subscribe so that the video does not get lost on your YouTube, and please do share the link to this video to your friends, to your teachers, to anyone who might benefit from my lessons. My name is Tumi Selane. This will be lesson 4 on the lesson series of the DNA Code of Life. Lesson 4 is going to be on the history of the discovery of DNA. It is going to be a very short lesson. However, because it is on your examination guidelines, it is important for you to be able to engage in as well. I am a teacher from Milner High School, all the way in the Northwest Province. I have decided to do the lesson series of the topic DNA Code of Life in a total of 7 lessons. We have already went through the first, second, and third lessons in separate videos. So, to build context on today and the future lessons and future videos, please go back and check out those lessons because the content is a build-up. So today we are in lesson 4 and we will be looking at the history of the discovery of DNA. On a separate video, we will be looking at lesson 5, where we will be discussing and dealing with content relating to the second nucleic acid, which is called RNA. We will be looking at the RNA structure. On lesson 6, which will be on a separate video, of course, we will be looking at protein synthesis. Now, this will be a 2-part lesson where on the first part, we will be looking at the process of of protein synthesis, which will comprise of transcription, movement of mRNA, as well as translation. The second part of lesson 6, we will look into amino acid coding. On the 7th and last lesson, we will be looking into DNA profiling, where we will be looking at the definition and uses of DNA profiling, and we will be looking at factors that affect the reliability of DNA profiling. We will be doing some interpretive work where we are going to interpret different DNA profiles, firstly to determine paternity, and we will be looking at DNA profiling as a way of forensically determining a possible criminal in a criminal case. Okay. For the context of this lesson, we will be looking at the historical events that led to the discovery of DNA as we know it today. We will be looking at the roles played by these 4 characters, Watson, Crick, Wilkins, as well as Franklin. Now, it is not important for you to know the social aspect of this history. All that we need to know are the different roles that are played by these people in the discovery of DNA. Okay, so you do not need to know the social aspect of these characters. We only are focusing on the scientific based roles that were played by these 4 characters in terms of knowing what DNA looks like and how it functions today. On this slide, we will be looking at the story of the history of the discovery of DNA. Like I said in the previous slide, the social aspect of this brief history of the discovery of DNA is not important. Things like the years and the places of occupation and the likes are not important. What we will be focusing on is the roles that are played by the 4 different scientists in putting forward the model of DNA that we know today. For the purposes of the lesson, I have sourced an extract which I found in page 8 of the textbook Understanding Life Sciences 3rd edition CAPS. Okay, now here is the extract. It is titled Brief History of the Discovery of DNA. So I will be reading through the extract, and I hope that you will be picking up some points which will be very helpful in the following slide when we complete a summary activity of the different roles that are played by the 4 scientists. Are we ready? Okay, let us start reading. The extract reads thus: no one has actually seen DNA or RNA. Our knowledge of the structure of DNA comes from a theoretical model put forward by scientists Watson and Crick. In other words, what we have studied so far is a theory of what DNA looks like. James Watson and Francis Crick teamed up and carried out their research at Cambridge University. At King's College London, 2 other scientists, Rosalind Franklin and her assistant, Maurice Wilkins, were also working on researching the structure of DNA. Rosalind Franklin realized that DNA was some sort of a double helix from X-ray diffraction pictures she took. Maurice Wilkins disliked Rosalind Franklin. He showed these X-ray diffraction pictures to James Watson. This helped Watson in his own research, especially about the helical structure of DNA. Watson and Crick continued their research and found that DNA contained similar amounts of adenine and thymine, and also similar amounts of cytosine and guanine. The similar amounts of adenine and thymine and also cytosine and guanine gave them the idea of complementary base pairing, i.e. that cytosine always pairs with guanine and adenine always pairs with thymine. Another significant aspect of the findings was that the complementary pairing of bases allowed DNA to make exact copies of itself. See DNA replication later. Actually, remember, we dealt with DNA replication in lesson 3 previously. Continuing reading the extract. In 1953, Watson and Crick put forward their model of DNA, which indicated that it was a double helix with complementary base pairings. Rosalind Franklin died of cancer in 1958. And in 1962, James Watson and Francis Crick, as well as Maurice Wilkins, who provided important X-ray diffraction pictures, were awarded the Nobel Prize for their work. If Rosalind Franklin was still alive, it is likely that she and not Maurice Wilkins would have been the 3rd person to share the Nobel Peace Prize with Watson and Crick. End of the extract. Okay, so from this extract, we can see that there are 4 different scientists that were working to explain what DNA looks like and how it functions. On one side, we had Watson and Crick, and then on the other side, we had Wilkins and Franklin. Okay, so it is actually Rosalind Franklin that came up with the pioneering research from her X-ray diffraction pictures. She discovered that DNA is some sort of a double helix, okay, from these X-ray pictures that she took. Now, she was working with Wilkins, who actually disliked her. It just goes to say that you can work with somebody and that they might not actually like you, but she did a immoral thing, might I say, or an unethical thing, actually. Not immoral, but unethical in terms of the principles of science, and took those pictures that were actually intellectual property of Franklin, and she took them to James Watson. And James Watson took that intellectual property, used it together with, from the intellectual property that was taken to James Watson, Watson and Crick continued with their own research, and from that basis they discovered the DNA structure that we see today. Okay, now this is a lot of information. It's a lot of drama for me. So let us go to the next slide where we are going to summarize this. In terms of the content of the history of the discovery of DNA, it is not a lot for you to know, but it is quite important because it is sourced in the examination guidelines, you guys. So, just to summarize all of that data from the previous slide, we are going to use a table to give a precise summary of the things that you need to know. So that instruction of this activity reads, complete the table below to summarize the role or roles of each character or characters played in the discovery of the DNA model. Okay, so on this side we have the characters: Rosalind Franklin, Maurice Wilkins, and remember, James Watson and Francis Crick worked together. So their role I have put in one column. Okay, now please pause, go back to the extract, listen to it again, and fill in the different roles that are played by these different characters. Okay, and before you look at my corrections, compare that to your corrections. And if you have any additional thing that you need to add, please do add that in the comments to this video. So, what role was played by Rosalind Franklin in the discovery of the DNA model? Simply, Rosalind Franklin discovered that DNA is a double helix from X-ray diffraction pictures she took. That's all that you need to know. So the pioneering research was done by Rosalind Franklin. She provided X-ray diffraction pictures that proved that DNA is some sort of a double helix. All right. Now, Franklin, remember, was not working alone. She was working with Maurice Wilkins. And then, what role was played by Maurice Wilkins? In terms of his role, he took the pictures and showed them to Watson without Franklin's knowledge or without her permission. Okay, so what he did, what Maurice Wilkins did, was actually quite unethical in terms of the ethics of science and research. Now, after showing Watson the pioneering research, what did Watson and Crick do after? Watson and Crick actually used Franklin's diffraction pictures to further their research, which ultimately led them to put forward the model of DNA that we know today. So they took that research and continued from that, and then they discovered, or they put forward, the DNA model that we know today. And it is they, together with Wilkins, that received the credit for putting forward the DNA model that we know today. Okay. Like I said, ladies and gentlemen, this was a very short and sweet lesson on the history of the discovery of DNA. That is the end of lesson 4. And if you liked this short presentation, please do like, comment, subscribe, and share the link to this video, because it might also benefit another learner who might want to succeed in life sciences and ace their final examination. Thank you so much for watching, and I'll see you in the following lesson, which is titled The RNA Structure. Goodbye.